The natural world teems with healing power. For centuries, we have looked to plants for medicines. Now a new era is dawning. Biotechnology is revolutionizing the way we discover and develop drugs. At the heart of this revolution lies a remarkable technique: tissue culture. This powerful tool allows us to unlock the vast potential of plants for human health.
Imagine taking a tiny piece of a plant, a single cell perhaps. Now imagine coaxing that cell to multiply, to grow, to form an entirely new plant. That is the essence of tissue culture. It's a delicate dance of science and nature. We provide the right conditions, the nutrients, and the environment. In return, nature works its magic, transforming a single cell into a thriving plant. The journey begins with selecting the right plant. This is not just any plant, but one chosen for its unique and valuable medicinal properties. The selection process is meticulous, involving careful examination and consideration of the plant's potential benefits. Scientists look for plants that have shown promise in traditional medicine or have been identified through research as having specific therapeutic compounds. We choose it for its medicinal properties. These properties are often linked to the plant's ability to produce certain bioactive compounds, which can be used to treat various ailments. The chosen plant is then brought into the lab where the real journey begins.
The next step is crucial: sterilization. This involves thoroughly cleaning and disinfecting all lab equipment and surfaces to ensure that no contaminants can interfere with the tissue culture process. Contaminants such as bacteria, fungi, or viruses can easily ruin the entire culture, so this step is taken very seriously. We must create a pristine environment, free from contaminants. This is achieved by using a laminar flow hood, which provides a sterile workspace by filtering the air and preventing any airborne particles from entering the work area. The lab is also kept at a controlled temperature and humidity to further reduce the risk of contamination.
Then, we carefully extract a small piece of plant tissue. This tissue can come from various parts of the plant, depending on the desired outcome of the culture. This could be a leaf, a stem, or even a root. Each type of tissue has its own unique properties and potential for growth. The extraction process is delicate and requires a steady hand and a keen eye to ensure that the tissue is not damaged, or even just a few cells. These cells are the building blocks of the plant and have the potential to grow into a whole new plant under the right conditions. This tissue is placed in a special nutrient-rich medium. This medium provides all the essential nutrients and hormones needed for the tissue to grow and develop. It is carefully formulated to mimic the natural environment of the plant, providing the perfect conditions for growth. This is where the magic happens. The cells begin to divide and multiply, a process known as cell division. This is the first step in the transformation of the tissue into a new plant. The cells begin to divide and multiply, forming a mass of undifferentiated cells called a callus. This callus is essentially a blank slate with the potential to develop into any part of the plant.
This callus has incredible potential. Under the right conditions, it can be induced to form roots and shoots, the basic structures of a new plant. Under the right conditions, it can be induced to form roots and shoots, which are the essential components of a new plant. This process is carefully monitored and controlled to ensure the best possible outcome, eventually developing into a whole new plant. This new plant is genetically identical to the original, making it a perfect clone. This is particularly useful for preserving rare or endangered species, as well as for producing large quantities of plants with desirable traits. This process, called regeneration, is a testament to the remarkable ability of plant cells to differentiate and specialize. It showcases the incredible potential of plant tissue culture in agriculture, horticulture, and medicine.
Finally, the young plantlets are carefully acclimatized to normal growing conditions. This involves gradually exposing them to the outside environment, allowing them to adjust to changes in temperature, humidity, and light. They are ready to be transferred to soil, where they will continue to grow and develop. This is a critical step, as the young plants need to establish a strong root system in the soil to support their growth, where they will continue to grow and thrive. With proper care and attention, these plants will mature and eventually produce their own seeds, completing the cycle and ensuring the continuation of the species.
Tissue culture is a game changer for pharmacognosy, the study of medicines derived from natural sources. This innovative technique involves growing plant cells, tissues, or organs in an artificial medium under sterile conditions. By doing so, we can bypass many of the limitations that traditional cultivation methods face. It allows us to produce large quantities of valuable medicinal plants in a controlled environment. This means that we can ensure a consistent supply of these plants, regardless of external factors such as climate or soil conditions. The controlled environment also allows for the optimization of growth conditions, leading to healthier and more robust plants. No longer are we limited by the vagaries of nature, the changing seasons, or the threat of pests and diseases. Traditional farming methods are often at the mercy of these unpredictable elements, which can lead to significant losses. With tissue culture, we can create a stable environment where plants can thrive without these risks. With tissue culture, we can have a steady, reliable supply of medicinal plants year-round. This is particularly important for plants that have a high demand in the pharmaceutical industry. By ensuring a continuous supply, we can meet the needs of researchers and manufacturers who rely on these plants for developing new drugs and treatments. But it's not just about quantity. The quality of the plants produced through tissue culture is often superior to those grown in traditional settings. This is because we can control every aspect of the plant's environment, from the nutrients it receives to the light and temperature conditions. Tissue culture allows us to enhance the quality of medicinal plants as well. By carefully monitoring and adjusting the growth conditions, we can ensure that the plants produce higher levels of the desired compounds. This can lead to more effective medicines with fewer side effects. We can manipulate the culture conditions to boost the production of specific compounds that have therapeutic value. For example, by adjusting the nutrient levels or the light exposure, we can increase the concentration of certain alkaloids or flavonoids that have medicinal properties. Imagine increasing the potency of a plant's anti-cancer properties or enhancing its ability to fight inflammation. These are just a few examples of the potential benefits of tissue culture. By harnessing this technology, we can unlock new possibilities in the field of pharmacognosy. Tissue culture makes this possible. It represents a significant advancement in our ability to produce and improve medicinal plants, paving the way for new treatments and therapies that can benefit countless people around the world.
Let's meet some plant stars who have benefited from tissue culture. These plants, through the marvels of modern science, have not only thrived but have also contributed significantly to human health and well-being. Take, for instance, the Madagascar periwinkle. This seemingly ordinary plant, with its delicate pink and white flowers, hides a powerful secret. This unassuming plant produces vital anti-cancer compounds, specifically vincristine and vinblastine, which are used in the treatment of various cancers, including leukemia and Hodgkin's lymphoma. Tissue culture has been instrumental in increasing the yield of these compounds, making life-saving treatments more widely available. By cultivating plant cells in controlled environments, scientists can produce these critical compounds in larger quantities and with greater consistency. Another example is Panax ginseng, a plant highly prized in traditional Chinese medicine for its energy-boosting and immune-enhancing properties. Ginseng has been used for centuries to improve overall health, reduce stress, and enhance physical stamina. Tissue culture has helped to conserve wild ginseng populations while providing a sustainable source of this valuable herb. By growing ginseng in laboratory settings, we can reduce the pressure on wild populations and ensure that this important plant remains available for future generations. Then there's the Taxus spp, commonly known as the yew tree. This plant is the source of the anti-cancer drug paclitaxel, also known as Taxol. Tissue culture has revolutionized paclitaxel production, making this important drug more accessible to patients worldwide. By using tissue culture techniques, scientists can produce paclitaxel in a more efficient and sustainable manner, reducing the need to harvest yew trees from the wild. These are but a few examples of the remarkable impact tissue culture is having on the world of medicine. From increasing the availability of life-saving drugs to conserving valuable plant species, tissue culture is a powerful tool in the fight against disease. By harnessing the power of nature, we're developing new treatments and improving the lives of millions. The future of medicine is bright, thanks to the incredible potential of plant tissue culture. As we continue to explore and innovate, who knows what other plant secrets we might uncover.
The path of scientific discovery is rarely smooth. Tissue culture presents its own set of challenges. Contamination is a constant threat. Finding the optimal conditions for growth and development can be tricky, and scaling up production to meet demand can be a complex and costly endeavor.
But the rewards far outweigh the risks. Advancements in technology, such as CRISPR-Cas9 gene editing, are opening up exciting new possibilities for tissue culture.
Imagine creating plants with enhanced medicinal properties, resistant to diseases, or capable of thriving in harsh environments. This is the future of medicine, and it's closer than we think.
As we look to the future, one thing is clear. Our planet's health is inextricably linked to our own. Tissue culture offers a sustainable and environmentally friendly way to meet the growing demand for plant-based medicines. By reducing our reliance on wild harvesting, we can protect biodiversity and ensure the long-term survival of these precious resources. Tissue culture is not just a scientific breakthrough, it's a beacon of hope for a healthier and more sustainable future.